Okay, welcome everybody to tonight's talks. But first of all, I would like to acknowledge and pay respect to the traditional owners of the land on which we meet, the Ngunnawal people of the Wiradjuri Nation. It is upon their ancestral lands that the Australian Academy of Science is built. As we share our knowledge, teaching, learning and research practices, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of country. So again, welcome. This is the second series in our Canberra Speaker Series, The Science of Us. And tonight we welcome two speakers who are going to tell us about medicine made for you. My name is Professor Simon Foote. I'm one of the conveners for this series, along with Professor Lois Salmonson, who's works in reproductive health. I'm thrilled to be here in the iconic Shine Dome to join you tonight, and I thank you for being here to support Australian science. As you know, this series will take us on a journey through our lives from conception right through to death. Slightly morbid, I think, but still, that's not written in the notes. We hope that you will continue to join us throughout the series as we hear from leading scientists in the areas of mental health, addiction, ageing, and more. So tonight's talk is actually being recorded and will be online in the coming days on the Academy YouTube channel. So with that all done, I'd like to introduce the first of our two speakers tonight, and that is Academy Fellow and President-elect of the Academy of Sciences, Professor John Shine. Professor Shine is currently Professor of Medicine and Professor of Molecular Biology at the University of New South Wales, lab head at the Garvan Institute of Medical Research. John is an icon for those of us who work in molecular biology. He started his career at ANU, doing his undergraduate and PhD here, and he's famous for the Shine-Dalgarno sequence, which is a very important part of RNA that allows protein to be produced. I'm sure many of us have actually heard of this. He then beetled off to the University of California at San Francisco, UCSF, where he was instrumental in cloning a couple of very important genes, notably insulin and human growth hormone. He shuttled a little bit between Australia and the US, but ended up in a biotechnology company in the US, which ended up being very successful. He came back to Sydney, and after a period as deputy director, was actually made full director of the Garvan Research Institute in Sydney, where he did magnificent things over a fairly long period of time. He's been chairman of the NHMRC, chairman of CSL, president of the Powerhouse Museum, and he's a well-known philanthropist, as you must realise, because there is no coincidence that you are actually sitting in the Shine Dome. He was made a Companion of the Order of Australia last year, which complemented an early award, and won the Prime Minister's Science Prize in 2010. So please join with me in welcoming Professor John Shine.
Thank you very much, Simon. Your introduction's longer than my talk's going to be. It really is a great privilege to be here this evening and to talk to such a group of interested individuals who understand the value of science, because it really has such a major impact on our lives and holds such promise for the future that it's wonderful to be here in the Academy and to have the opportunity to talk a little bit about it. What I'm going to do today, I hope, is talk about medicines made for you and me, largely from the point of view of all we know about our genes, our genomes and genetic engineering, and how that sort of science around our genetics is really transforming healthcare. And it really promises a whole new sort of scenario for the future, for future generations. So I'll bore you just for a few minutes with a little bit of background to make sure we're all well aware of what we're talking about with DNA, our genes. As we all know, if I can get this to work, I'm not sure how I can work the little arrow. It doesn't matter, you can see it all on the slide. Our DNA, or our genes, is composed, the blueprint for life, is composed of 4 simple chemicals, just 4. Abbreviated G, A, T and C, guanosine, adenosine, thymidine, cytidine, but they're 4 very simple chemicals. They're arranged like beads on a string, and our DNA, our genome, has about 3.2 billion of those GATCs, like balls on a string. So you can imagine, if there's only 4 different chemicals, but there's 3.2 billion of them, the permutations that are possible are enormous. GG, AT, CC, etc., etc. Each cell, each of the 75 trillion cells in our body, every time they divide, they have to replicate exactly that 3.2 billion sequence of GATCs. And normally that's incredibly efficient, and they don't make many mistakes. Occasionally a mistake is made. If that mistake, for instance, a G is put in where perhaps a C should be put in, if that mistake is in an important gene that controls a function in the body, it's called a mutation, and it may or may not have bad consequences. On the other hand, if it occurs in a part of the genome which isn't coding for any particular important gene, it's just called a polymorphism, and it's part of the difference between all of us. On the slide there, for those of you that are very astute, you'll see there's a little red G and a little red A. That's to illustrate in this particular genome sequence, if that was mine, that might be those 2 things might be the very subtle difference between myself and Tom Cruise, and everything else remains the same. But there is a difference between me and Tom Cruise, but it is subtle. But if we go back about 20 years now when the human genome sequence was first being developed, the United Nations put out a declaration on human genome sequence and human rights. And I think it really encapsulates it well, although there's lots in the United Nations I disagree with, but it says the human genome underlies the fundamental unity of all members of the human family, because it is our common DNA blueprint. But as well as that, it's the recognition of their inherent dignity and the diversity, because of those little differences between me and Tom Cruise and between all of us. It does embrace what we are, but it also embraces our differences. So if we look again at our 3.2 billion GATCs, just to give you a feeling for the enormity of the information there, if someone here was prepared to donate their body to science and we homogenised them up and extracted their DNA and stretched their DNA molecules out into one single long strand, it would reach to the moon and back 8,000 times, just to give you an idea of the complexity. If we typed out anyone's human genome sequence, GATTCC, in the font size of the Canberra telephone directory, it would occupy 500 200-page directories. If I was to read out your individual genome sequence tonight at the pace of AT, C, TT, GG, CT, I'd be talking to you for 100 years non-stop. I'm obviously not going to do that tonight. But that's just to give you a concept of just how much information is there. So what do we already know about the human genome sequence? It's often called the Book of Life. It's actually 4 books. It's a very sophisticated history textbook because it teaches us about human evolution and human migration around the planet when we look at the differences between different ethnic groups, for instance. It's also a very sophisticated construction manual and a parts list because within that GATC 3.2 billion are all the information, all the blueprint, everything that is, all the information that's required to take us from a single fertilised egg to a complex human being. It's also a medical textbook, of course, because within that sequence it has all of the information about what goes wrong in different diseases, what puts us at risk of particular diseases. And it's also a who's who in the world, because although I joked about my GC difference from Tom Cruise, it is a blueprint of us as an individual. So it contains an enormous amount of information. We know, as I said, it's 3.2 billion base pairs long, but that enormous amount of GATCs only encodes about 30,000 genes, as we call them, the functional units that contain the instructions to make enzymes and hormones and the building blocks of the human body. If you do and each gene's about a few thousand GATCs long. So if you do your mathematics, you realise that actually only a very small percentage of the human genome actually encodes these functioning genes and proteins. The other 90-odd% has been so-called junk DNA, and it's arisen because during evolution, as we've evolved from a simple organism to a more complex organism, we've done that by bringing together genes and DNA from different areas, and there's a lot of stuff in between that's been like the joining stuff. Now, we now know that a lot of that so-called junk DNA has a function, and it actually switches on and off genes in different ways, and there is another complex level of regulation there. But nevertheless, a lot of it is still so-called junk. And we know a lot of it has arisen from various viral sequences that have been inserted during evolution into our DNA, and they've been kept there in an increasingly degenerate form as we've evolved, but nevertheless it's just basically random junk. But it's also the fodder for future evolution, because changes or mutations in that DNA might, by chance, with natural selection, give rise to new functions and hence evolution of a species. We also know that this slide's a bit out of date now, it's closer to 300 human genes are more similar to bacterial genes than they are to other higher organisms like yeast and worms and flies and plants. Most of those genes encode important enzymes necessary for metabolism and energy production, so they're critical human genes, but they came from bacteria originally. So the concept of swapping DNA or genes between species, the concept that that only occurs with mad scientists in the lab, and it's a terrible thing, is ridiculous. It's natural. It occurs throughout human evolution. DNA is switched around. So we've learnt that. We've also learnt, if we look at the mutation rate that occurs in the DNA in sperm versus the mutation rate that occurs in the egg cell in women, that in fact the sperm mutates at about twice the rate of eggs. That allows me, as a male scientist, to point out that this is a definitive proof that men are the drivers of evolutionary innovation. When I tell Carola that, she says, no, it just means men are the source of human error, of genetic mistakes. Any way you like. So we are all about 99.9% identical. Our DNA is about 98% identical to chimpanzees. But that 2%, of course, is very important, and it regulates in complex ways all the different genes. The point being that we're 99.9% identical, so we are all very similar as human beings. But of course, 0.1% of 3.2 billion is still several million differences between us. And it's those differences between us that medicine is beginning to exploit to understand how we can tailor particular ways to prevent disease and treat disease to the individual. And that's what I suppose tonight's mostly about. I want to make one other point that shows how the rate of acquisition of this new knowledge of our genes has been exponential over the past 20 years. And I also want to stress the importance of what's called bioinformatics, and that is having the computing power to analyse those genes and what they mean. There's not much point in me reading out for 100 years GATCCC if it doesn't mean anything. We have to have the computing power to analyse it, to compare different individuals, different populations, those that are sick, those that aren't sick with a certain disease, and find those differences. This is just a graph that shows you the cost of doing the complete human genome sequence of an individual and how rapidly that has come down as the technology develops. The straight white line there is Moore's law, which is that computing law that says the number of transistors per square inch doubles every year or something. The computing power continues to grow exponentially like that. But you can see in the green line, the cost of doing a human genome sequence has been even more dramatic. And if we look now back in the year 2000, which to me wasn't that long ago, but I suppose it's 18 years ago now, when the first human genome sequence was announced with great fanfare by Bill Clinton and who was the guy in the UK at the time? Anyway, the UK Prime Minister. It cost at that point $3 billion and 15 years to get to that point. 2011, The technology had developed such that it was now 2 months and about $20,000. Today, we can take a blood sample from anyone in this audience and do your complete 3.2 billion DNA sequence in certainly less than 2 days, and it costs about $500. Nevertheless, it still costs about $1,000 or so to do the analysis of that sequence with computers. But nevertheless, the point being that the cost of understanding your exact human genome sequence is now down at the level of a standard diagnostic test for many diseases. So that's now going to be something which permeates the whole of medicine in the future. How do we use this new knowledge when we talk about medicine for you as an individual? First, we have to, of course, to realise that most of the diseases that challenge modern society are what we call multifactorial diseases. They're due to the interaction between various environmental factors and our inherent genetic makeup, genetic factors. We know already about many diseases and the environmental factors. Heart disease is a good example. We know high cholesterol, obesity, high blood pressure, smoking, all these things predispose you to have an increased risk of cardiovascular disease and having a heart attack. But we also know of examples where you have a healthy 40-year-old jogger running around in the park all the time, skinny as a rake, you know, eats all the right sort of vegetables all the time, drops dead of a heart attack at 40. On the other end of the spectrum, you have a gentleman who goes to McDonald's every day, dies in bed with his fourth wife, smoking a cigar at the age 90 when he has a heart attack. The difference is the genetic makeup in that situation. So we need to know the genetic as well as the environmental factors to practice true preventative medicine. And that applies to all of the major diseases that challenge us, as I said today: cancer, heart disease, asthma, osteoporosis, dementia, obesity, diabetes, mental illness. All of these diseases have environmental and genetic components. And now we're beginning to understand in great detail the genetic components so we can do something about the environmental components. First of all, if we understand the genetic components, we can make new drugs aimed at the product of that gene. But secondly, even today, we can practice prevention far more effectively. If I use that same example of heart disease, if you go into a doctor's surgery and there's 6 adult males there, and the doctor says, look, if you 6 guys don't stop smoking and get your cholesterol down and all, you will have a heart attack by the time you're 60. What happens? They all look around each other and say, it'll be him, not me, you know. It's a generic thing. If you take a blood test and say to each of those individuals, you have a genetic mutation in your apolipoprotein X gene, which predisposes you to having a heart attack with fair degree of certainty, unless you do something about the environmental factors, then they stop smoking and they lose a bit of weight, etc. So you can use that information already. Let me give you 2 quick examples of how these complex diseases, genetics and the Human Genome Project can help. Cancer is obviously a major one. We all know that cancer is a progression of mutations of different genes going from a normal cell that divides slowly only when it should, all the way through to an aggressive cell that's growing and dividing all the time. And there's a stepwise pattern of mutations in different genes. An analogy to perhaps think about is the accelerator and the brake on a car. And there are a set of genes that we have which act like accelerator genes. When they're turned on, they tell the cell to divide. There's a set of other genes which, when they're turned on, they tell the cell to stop dividing. They're called oncogenes and tumour suppressor genes. If you have a mutation in one of those accelerator genes, it's like sitting in the car, you put your foot on the accelerator. As long as your other foot's on the brake, the car might move forward slowly, but it won't take off. But of course, every time the car moves forward a bit or the cell divides a bit, there's another chance of another mutation happening. And so if you get another mutation, and this time it's in one of the brake genes, it's like taking your foot off the brake a bit. And your other foot's just on the accelerator, the car starts to go faster. And there's a cascade of mutations. A few more accelerator genes go, a few more tumour suppressor brake genes go, the cancer starts to take off. So knowing that, if we can look at any individual cancer biopsy and look at those genes and say, what genes have mutated that are driving this cancer, then you've now got a target for the development of new drugs, or you've got perhaps a new drug that already targets the product of that gene. So you can personalise that treatment. Rather than saying, you've got cancer, hit you with a horrible chemical that kills every cell in the body, maybe kills the cancer a bit more than the normal cells, and hope for the best. So it's becoming far more precision, far more personalised. Look at another area close to my heart, obesity. We all know we have a certain set point in our body weight. And some individuals, although they do everything they can with environmental factors, still have real trouble controlling their body weight. Others have no problem. We now know, and this is a simplistic slide, but our body weight is set by a complex feedback mechanism. Up in the brain, you have a series of chemicals that are released which stimulate feeding behaviour. They stimulate your appetite. They stimulate your preference for certain sorts of foods, whether it be fat or carbohydrate or protein. Those, when you then eat stuff, of course, like that, you release all sorts of other hormones, the end result being that you lay down fat in a fat cell called an adipocyte. And the more fat in that fat cell, the more this OB gene, from the Greek meaning, it's an obesity gene, it produces the hormone leptin. And leptin, from the Greek leptos, meaning thin. So the more fat in the fat cell, the more the OB gene turns on and makes more leptin. The leptin goes into the bloodstream, goes up to your brain and tells it to shut off or not all these other hormones which are driving your appetite and your metabolism. So we've all got this circuit going on. And it's an incredibly important one because surviving back in our evolutionary times, in caveman times, we really had to make sure we effectively used every bit of food we could get. So there's all sorts of mechanisms here to ensure that. However, if I happen to have a slight mutation in my neuropeptide Y gene in the hypothalamus, so I don't make it quite enough of that, then I may eat more than I should and I'll put on more weight. Or if I have a mutation in my leptin receptor, I won't recognise the signal from the fat cells telling me to go easy. So you can target the drug to the individual's particular type of obesity in a way that would be effective for them, not for the general population. Just trying to give you two different examples. The other thing all this knowledge is doing, it is changing the way we do medical research. And I'd just like to give you an example, for instance, prostate cancer. Medical research in general has always been driven throughout the ages, and still is in many ways, by what we call hypothesis-driven research. So you have an idea or a theory or there's some experimental evidence, and you hypothesise that, if we use prostate cancer, prostate cancer cells make more of a certain growth factor, therefore this growth factor may be driving the prostate cancer, therefore a drug against that growth factor may work in prostate cancer. That's the hypothesis-driven way. What's happening now, because of our ability to do this sort of analysis, is you look at normal prostate tissue and a prostate cancer tissue shown here from a variety of individuals with prostate cancer, and you look at all the different 30,000 genes that we've got in our genome, and you say which ones are turned on when they shouldn't be. And you can see this particular gene, as shown by that graph there, says in normal prostate cancer, this gene's not turned on at all. But in all prostate cancer samples, it's expressed at a high level. Therefore, this is a great target to develop a drug against to treat prostate cancer. But then it becomes even more individualised. If I show you another one for breast cancer, same sort of thing, different gene, of course. In the green on your right, a series of individuals with normal breast tissue. This particular gene just doesn't turn on, it's not involved in development of breast or breast tissues. However, you've now got a series of breast cancers where this gene is turned on in some breast cancers but not others. So a drug based against that particular gene would be exactly what you want to treat those individuals' cancer and not the other one. Simon's telling me all my time's running out, I think. So it's all about prediction, prevention and individualisation. I'll just skip through a couple of these slides. I just want to make a couple of points though, Simon, and one is that although this is all new and sexy and wonderful, it's not all that new, in that traditionally we know family history is important. If your parents died of a certain cancer, there's an increased risk you might die of that cancer. We know behaviour is important. We know environment's important. Social circumstances are important. What this is doing, this technology, is just allowing you to be far more specific and detailed at the individual level rather than that sort of class level. It gives us safer, more effective drugs. I want to make two other points quickly. as well as personalising the treatment of our different diseases, it has enormous impact in infectious disease. New bugs, viruses, bacteria are evolving all the time to threaten us. What we can now do, as soon as a new flu virus occurs, as soon as a new other sort of virus, as soon as a new bug occurs, within basically a week, you will sequence the genome of that new virus or bacteria and develop ways to think of antibiotics to treat it. We'll never win the war against infectious disease because there's a constant evolutionary battle, but we will more and more win each of the battles much more rapidly. So it has a big impact on infectious disease. The other area which we're not talking about is stem cells, which is also, together with the genetics, is revolutionising how we treat individuals. So beyond 2018, all human genes have been cloned, have been sequenced. There's now these very extensive genomic databases. Hundreds of thousands of people's genome is in a database that anyone can look at, and those individuals, you know their clinical history, have they had this disease, have they not had this disease, and it's an enormous amount of information there. So we're beginning to understand this matrix of genes that are involved in all these different multifactorial diseases, leading to a whole new range of therapies and a lot of emphasis on preventative strategies. So we're going to get a rapid information on a lot of information about individuals. But there's an increasing gap. These are the problems now. There's an increasing gap between what we know how to diagnose and what we know how to treat. And a good example is a horrible neurological disorder called Huntington's chorea, where we can do a simple blood test with everyone in this audience and do a quick genome test and say, you've got that mutation, you will get Huntington's by the time you're 40. Maybe it's a bit too late for some of us, but you will get Huntington's by the time you're 40. But there's absolutely nothing we can do about it, because it's a necessary step, though, to know that mutation and what it means in order to develop a treatment. And we're in that frustrating period with many of these disorders. And there's often we tend to think we know everything, but of course we don't. There's a whole bunch of social issues. Look, I'll just touch on one. Most of them are normal social issues brought into sharp relief by this new technology. And a good example is this human responsibility. If you're arrested in a bar one night because you beat someone up really badly in a totally unprovoked way, and you go to the judge, you go to court the next day, and you've probably got a tie on, you look a bit different than you did in the bar, but your defence lawyer argues, Your Honour, my poor client, he had a terrible family upbringing, he was abused, he was all this sort of stuff, he's been psychologically scarred. It's not his fault. And often that mitigates the sentence to some extent. Now, you can go in there and say, not my fault, I've got a G to A mutation in my neuropeptide Y receptor gene that makes me uncontrollably aggressive in certain circumstances. It's not my fault. Now, society has to deal with that sort of increased information as well. So there's a lot of issues that we need to do. Just finish the last slide, Simon. I just think, for me, this is a bit that summarises it all, and that's the saying that imagination based on knowledge is the key to discovery. Humanity has always been rich in imagination. We are now in a period of enormous exponential increase in our knowledge base, and that really is meaning we're in a real period of discovery. Every day there's a new discovery about how our bodies work on an individual basis and what we can do about it. Later during questions I can tell you something very interesting about environment-gene interactions, but I've run out of time, so thank you very much.
Well, that was absolutely fascinating. Thanks very much for that, John. Now, it gives me great pleasure to introduce tonight's second speaker, who is Professor Carola Vinuesa, Professor of Immunology from the John Curtin School at ANU and Director of the Centre for Personalised Immunology. Now, Carola, as you will see very shortly, is Spanish. She did her early education in Madrid. She did her medical degree in Madrid and then moved to Britain, where she actually did her specialist medical training and also a PhD at the University of Birmingham. So we're very lucky that Carola made a decision at that time to concentrate more on her research than on her clinical medicine because she is at the forefront of taking many of the ideas that you've just heard from John and translating them into actual therapy for people with disease. As well as that, she does some very exciting fundamental research into immunology. She is a recipient of the Prime Minister's Prize in Biological Sciences in 2009. She's a fellow of the Academy, and Carola, I'm looking forward to hearing your talk.
Thank you, Simon. I'm not sure you will see that I'm Spanish, but you will hear that I'm Spanish. I have not managed to get rid of my accent despite 25 years living outside of Spain. And I was going to say before starting that it was going to be a very hard act to follow John Shine at the Shine and Dome. My daughters were very impressed with that. But actually, and don't take me wrong, John, it's actually a very easy act to follow because you've given such a fantastic overview that I think you've made my job very easy now, going through what I was going to tell you. So I'm going to tell you the first part of my talk, a little bit of the kind of things that we are doing in precision medicine, but then I thought I would take the chance and the opportunity to tell you about other things that we are now being confronted when we look at our genomes. And there are issues that we don't tend to think about often as clinicians or scientists, but that are increasingly important, and we have to provide some type of forum for reflection. So precision medicine, as John has told you, is about identifying gene or variation that can actually help us tailor drugs better to each individual patient. And apart from cancer, which John has talked about and has enormously benefited already from precision medicine, the next area that arguably is benefiting most is the area I work in, the immune system. And this is because of three reasons. One is that we know in exquisite detail hundreds of molecules and many cell types that work together in many different pathways to orchestrate an immune response. Second is because the immune cells are very easily accessible. We get them from the blood. When we bleed a patient, we can immediately work with them and validate that the functions, the genetic mutations that we have found are functional. And thirdly, because in immunology, like other areas, we've got a very vast array of therapies or drugs already on the shelf that we could use. Many of them have been developed for other purposes, but if we actually know the mutation in the patient, we do have an indication, and we could go into treatment quite rapidly. Now, not surprisingly, we have discovered over 300 genes already that cause immune deficiencies in particular. Immune deficiencies are diseases in which, for example, kids are susceptible to different infections and become or get these recurrent infections against, from viruses, bacteria, worms. And this field has been, as I say, very relatively straightforward to find the mutations that cause disease. Then we come to the area that I work in, which is a little bit more complicated, is the area of autoimmune diseases. These are diseases in which our own immune system fights our tissues and organs, and there are over 80 of these diseases. We understand them poorly, and there are no drugs for any of these, really. There are no cures. We manage them as best we can. And part of the problem is that these diseases are very heterogeneous. Here you have two girls, and they are in the form of cartoon, but these are two real patients that we've had. You can see Julia on the left, and it's not her real name, but she's presenting with a type of lupus that mainly manifests as having joint and brain problems. Whereas on the other side, you've got another little girl, we call her Carmen, and her type of lupus is mainly presenting with problems in her kidneys and heart. So you immediately see that this disease is probably a group of different diseases caused by different molecules and pathways. But at the moment, we don't really know what they're going to respond to, so what we do is just treat them blindly with drug after drug until, if we are lucky, one responds. And you know that some of these treatments can be quite toxic, they can be quite expensive, and in many cases, they still fail to treat the most serious manifestations of disease. So, thanks to this incredibly blue sky discovery of a completely new form of sequencing that John has talked about, next generation sequencing, we can now do what it was unthinkable some years ago, which is going to sequence that genome, find that particular letter change that can tell us what the actual specific disease Julia has, and that's what we've done. And if we find a gene that we have a bit of an idea of how it works, we can actually test it in the laboratory and test whether that particular mutation actually makes the gene not work properly. And this was the case of Julia. She was diagnosed when she was only 2.5 years ago with a disease called lupus, and it was a very severe form, and at the age of 4, she developed a brain infarct. And sequencing her genome revealed a homozygous mutation in a gene that we call TREX1. We functionally validated that this gene was causing the disease, and we now understand how the gene works. If it's mutated, it causes the accumulation of a cytokine that we call a type 1 interferon, and this in itself causes a big part of the disease. And now there's a drug in clinical trials that specifically blocks this aberrant production of type 1 interferons. Now, the drug wasn't being developed for lupus, it was developed for another disease, a disease of the central nervous system, but we have hopes that it will work in cases like this. Now, there are other cases that are a bit more complicated, like the one of Carmen. We found the gene, we were excited, but we don't quite understand how the gene works. So it's not so easy to validate, and the question is, could we have a mouse model where we could test and try and understand how disease comes about? Well, again, thanks to another blue sky discovery, and I think it's always important to emphasize the importance of blue sky research, we now have a method available to us in which, in a very cheap and fast way, we can change any letter in the genome by cutting the DNA and substituting it by another letter. This technique is called CRISPR. It was used by bacteria to defend themselves against viruses, and we have now applied it to edit genomes. So we can edit the genome of a mouse and introduce the precise point mutation that Carmen has. And in fact, that's what we've done. And it has been very exciting because now we have a mouse model that develops the same type of disease as Carmen and as probably many other humans around the globe that have lupus. And to my mind, this is probably one of the best, if not the best, mouse models of lupus, because it's not a spontaneous random mouse model that appeared in nature that might be telling us relatively little about human disease. This is a model that is telling us how disease develops in a patient. So we can use the model not just to understand how that gene causes disease, but now to also trial drugs that we have already, but we just don't know to which type of drug Carmen would respond to. So this is very exciting, and it's the basis of a PhD project in our lab at the moment. Now, it can get even more complicated. I've told you the cases of extreme, early onset childhood diseases, which tend to be more severe, and because of that, sometimes they are what we call monogenic. They are caused by a single gene. But some people, it's not as simple as one gene explains the disease. There might be mutations in 2 or 3 different genes. And then on the other hand, also the chance that any 2 patients with disease have the same combination of mutations is quite rare. So what can we do about this? Well, by dissecting these very extreme phenotypes, like the one of Julia and Carmen, and by generating these mouse models, we can then interrogate them for what we call biomarkers or signatures, for example, in the blood, that tell us something about that particular pathway of disease. So, for example, a substance that's increased in the blood in the case of a disease caused by the gene that Julia has. And with those biomarkers, we then have clues. We have some type of idea of what we should be looking for in the general population. So we can then screen larger cohorts without the need of sequencing their entire genome and start dividing them into groups, stratifying them according to the biomarkers. So then we can speculate that perhaps some of the drugs that work in those mice models that represent each of those pathways might work in these cohorts. And that is what we are doing at the Center for Personalised Immunology here in Canberra, which is funded as an NHMRC Center of Research Excellence. We now can start from the patient, sequence the genome, identify the mutation. In many cases, we can introduce it in cell lines or mice, use those models to understand how disease develops, to find which are the best biomarkers, test drugs that we think might be useful, and eventually come back to the patient. And obviously, it's quite more complicated than this, and it doesn't always work, but we are learning things about immune diseases. And then, of course, we take this information into larger cohorts, and, as I say, use this knowledge to find groups that might be responsive to a particular gene, to a particular therapy. And on the basis of the work of Canberra, of the Center for Personalised Immunology, ACT Health was quite visionary in funding a center here in Canberra called Canberra Clinical Genomics that we've built up based on the pipeline of the CPI. So we are now extending genomic medicine to other clinical specialties, particularly those cases that we think would benefit from genetic therapy. And overall, with the effort of CPI and Canberra Clinical Genomics, we've now sequenced over 1,200 genomes. And we have had some good stories, and this is just an example that appeared in the Canberra Times a few months ago. This was a young woman with a severe inflammatory disease that causes inflammation in different organs, like skin and gut. It's quite rare, and thanks to finding the precise genetic cause of the disease, this lady was able to, or there was a change in her treatment that allowed her to get pregnant and have a baby, which was very special for her. I want to tell you about other diagnoses that this is making possible, and also the implications that it has in a patient's life to reach a genetic diagnosis, which not only is being able to start personalized therapy, but how important it is sometimes to simply reach that diagnosis. And this is a case, for example, of a girl that started feeling unwell when she was quite young, and it took until she was 21 years of age to get this definitive diagnosis. And I don't want you to read all of that. I just want to say that she really has had quite a number of problems and medical conditions that have caused quite a bit of, stress and disruption in her life. And with genome sequencing, within a few weeks, we could find this mutation that we knew and had been previously shown causes a particular disease. This is a mutation in an immune regulator, CTLA4. And the beauty, and this is what I was telling you, is that there happens to be a treatment that had been developed for a different purpose that specifically targets this molecule CTLA4. So this young lady could be started on treatment, and she's already feeling better. And we had a very similar case of a child. This one was referred to us from Spain. Again, 20 years without diagnosis, started his life with type 1 diabetes at 15 months of age, and quite a long list, and I've only put there a few of the clinical problems that he has had. He's currently very weak and with chronic gastrointestinal problems, besides quite a few other issues. Again, another mutation in CTLA4, now going to be treated differently. So we now have become a bit of a referral center. We are receiving samples from over 20 countries, and we are helping diagnose cases in quite a few countries in Eastern Europe, but also in other places like Oman, Turkey. We've started a center in China as well. And I want to emphasize that, this is an area where medicine is clearly being transformed, and it has become so transformative that we can say that through genomic medicine, the activities of clinical practice and research do actually merge into one. So this emphasis in translation is somehow diluted because we don't need to translate. We are already making difference to individual patients by doing the research on the actual mutations. So I just want to say that this also immediately tells us the importance of working very closely between the healthcare provider and the research centers. And in Canberra, we are quite fortunate because there is one main healthcare provider and two good universities. So there is obviously enormous benefit at doing this work in an integrated way. And then I want to move on to tell you some of these issues that we think a little bit less about, but are equally important. And as clinicians and scientists, we are driven by our passion to cure people and to just deal with finding treatment straight away. And there are quite a few things that we need to think about. So the first one I would like to put to you is, okay, so what are the diseases that we need to think of preventing or treating? And do we all think that the same diseases are really diseases? And there's a few nice examples and there are increasingly examples like this. And I'll tell you one about a deaf couple that turned up into an infertility clinic. And they basically wanted to have a deaf child. And eventually they got sperm from a family with 5 generations of deaf children, and they had the deaf child. And this provoked a little bit of the shocked parts of the community. But of course, their argument was that, in a deaf, proud community, being deaf is somehow a cultural identity, not necessarily a disability. And there are many other cases. There are cases of people with short stature, dwarfism, that actually do want a child that is also short. And this takes us to other area, which is now we are facing this crystal ball, with the information that our genome provides us. And now we can see into the future and potentially see what other diseases are we going to be susceptible with. And there's a few things that we have to bear in mind. First is that this science is still very imprecise. You know, there are a few issues. First, especially when we do this in a form of direct-to-consumer testing, there are mutations that are false positives. We don't know what filters might have been used. But also there are many variants that we don't understand the significance of. Even if they are in genes known to cause disease, those variants themselves might not be known to cause disease. And unless this is done in a clinical setting, it can create quite a bit of anxiety or even give false hope to patients. So we have to be careful and be conscious that we have to explain and interpret the results to people, and particularly remember that some of these results are then reported back to family members. So we have to do this properly. Also, you know, there's the question of, do we want to know about all diseases? I mean, there are some diseases in which, or some mutations that we know will, as John has mentioned, might tell you that you're going to develop dementia, but do we want to know that we are going to develop a disease that we cannot cure? And this is particularly relevant for mental health. Another issue is we are sequencing a lot of children, but we are not consenting children. And is this okay? Is this okay that the parent, you know, takes somehow a little bit over the autonomy of the child? I mean, would you like, or would I like to have my parents know about a disease that I'm going to develop? And are they going to treat me differently because of that? Are they going to be telling other people? So quite a few issues around that as well. Then the area of pre-implantation genetic diagnosis. Now, if you are wealthy enough and you can afford it, you can choose the embryo that you are going to implant on the basis of genetic testing. In Europe, there is a list of 54 diseases that are now tested as part of this pre-implantation genetic diagnosis. And we could argue that we are going to be confronted with difficult decisions. Many think that the gay gene is going to be discovered very soon. This is just an example. And you know, this is, I mean, many of us think it's unthinkable you would act on the basis of a gay gene, but James Watson, you know, the discoverer of the DNA helix, did say, well, if a woman, when the gay gene is discovered, if a woman finds that the fetus has a gay gene, she should be free to terminate the pregnancy. But, I mean, some of the decisions that might be a bit more straightforward are diseases that we're all familiar with. And everyone in this slide has one of these diseases that, you know, are in lists of pre-implantation genetic diagnosis. Steve Hawking had amyotrophic lateral sclerosis. Einstein had a form of high-functioning autism. Rita Hayworth had early onset dementia. Chopin, it's thought that he probably suffered from some type of cystic fibrosis. Lincoln had probably Marfan's disease. And Angelina Jolie had the genes that predispose to breast cancer. In a recent interview in the US, 10 to 15% of healthy individuals said they would terminate a pregnancy if their child carried these same genes. So with this, this takes us, you know, when we talk about cure and preventing, it takes us to this more slippery slope of the possibility of enhancement. No, if we can choose embryos while we find some of the traits that might be disadvantageous, we will know of others that could enhance performance, and this is this new type of liberal eugenics that is, you know, state neutral and non-coercive. And, you know, some have argued, and here I like the view of this American philosopher, Michael Sandel, that says that the problem with enhancement is not necessarily the perfection that it seeks, but how it would transform, you know, the values of our society. If you for example, I mean, he argues that there is a lot to be said for just being open, or the humility that it takes, and the enormous sympathy to be open to have a child that, you know, you might not be, it's something that you haven't bid for, that this unconditional love of you know, being open to any type of child that you might have. And that doesn't mean that you are not going to treat disease and make them better. And then finally, the idea of, I mean, of solidarity. If every human being is born equal, then, you know, there is an increased sense that we all have the same chance of suffering from unexpected disease. And this makes us all be a little bit more conscious of helping those that might be a bit less fortunate. So, and a good example is health insurance. No? In health insurance, for example, the healthy subsidize the unhealthy. But the moment that a population is conscious that they might be less prone to genetic disease because of some type of design, then they might not take up insurance and they might not try and share in the same way. So this could really transform our society. I'm going to go by these slides because I see that Simon is waiting there. I was just going to tell you about genomic editing, but I think it's going to be a bit too dense. But I will tell you that the last area that probably is a little bit worrying is equality. We know that even though prices are coming down, the possibility to sequence genomes and the infrastructure required is really only available to wealthy countries. And what could happen if we start taking genetic diagnosis and decisions that could be transmitted to future generations only in parts of the world, and you could envisage that eventually, you know, if we eradicate certain genetic diseases from certain populations, we could create perhaps two types of population. And, you know, this might really be some type of more real foundation to racism because there would be real genetic differences. So we have to be mindful of how we are using these technologies and the differences it might create. And then finally, you know, humankind has evolved under the pressures of, you know, external conditions. We have adapted by natural selection. And if we now enter into an era where we ourselves have the ability to modify the genes through, for example, genetic editing, we are going to be potentially changing, no, the ability to adapt. So this is something that, again, will have consequences at some point if we don't think carefully about it. So with all of this, I want to just thank everybody that, I mean, it's impossible to list everybody, so I'm sorry, but people in my lab and people, I mean, I would like to mention Todor Arsov, who has really been educating me in the importance of genetic counseling and some of the issues that we've talked about, the funding, and of course my daughters, who I definitely feel complete unconditional love, even if at days they are a bit grumpy. Hello. Okay, so thank you very much.
So Carola, thanks very much. You've taken us from a completely new paradigm in treating human diseases to an exercise in thinking about some of the complications. So we'd now like to invite